All right, let me just switch those settings. So we're going to take a peek at pharmacogenomics. I do want to start at the beginning. So we're going to define what it is. We're going to describe what those profiles may look like, and then identify some of the areas where the results may make a difference. So how can we use that information in the care of our patients? So what is pharmacogenomics? Very large word. We got to start with the DNA. So we're looking at our genetic profile. So for those of you non-science folks, hopefully this is a refresher and not too painful. So genetic content is stored within the body in the form of DNA, so deoxyribonucleic acid. So the DNA is actually a bunch of small pieces. So it's a phosphate group, it's a sugar group, and then it's one of these base pairs. So the different base pairs link up, and they're the code that encodes all of who you are and what you're able to do. For context, the DNA that you have in your body, if it was to be stretched out, it would be approximately 6 feet long. So we all have about 3 billion base pairs, and it's what the actual sequence is that makes a difference and how we see some of those differences. Now, what might that sequence look like? So this is just kind of trying to zoom in a little bit. Those base pairs link up. A links up with T, and then C links up with G. And the order that they're in tells your body everything that it's supposed to do. It is the massive instruction code. That code gets separated and then it gets repeated. So we make it into smaller messenger codes that then can be created into proteins, enzymes that make your hair and your nails and your saliva, all of your digestive enzymes, enzymes that make all of the proteins that make up all of the cells of your body, your neurotransmitters, the receptors, the enzymes that break those down. So it really is the instruction manual for how the entire body works. Now, there was a massive effort in the late 90s to early 2000s to map this, because we thought as soon as we knew what that code was, everything would be unlocked and we would have a lot more information. Well, we do have more information, but it wasn't quite as straightforward as we were hoping for. We thought if we knew the whole code, we would be able to say, this is what you're going to have, this is what we should use to treat you. So the whole code is the genome. That was way more information than what we could do anything with. As scientists continued to evaluate, they found that all of that genetic information is actually repackaged. So it's repackaged into chromosomes. So humans have 46 chromosomes, and they're made into these little, it almost looks like big X's, and they encode for different parts of our information. The reason that it's packaged this way is because if it wasn't packaged and condensed like that, it would be way too big for all of your cells to be able to hold it. So this helps make it nice and efficient. It reminds me of those vacuum sealed packages where you can suck out all of the air in the sweaters that were supposed to be this big or are now only that big. So it's a lot like that, like vacuum sealing your whole genetic code. Within each of those chromosomes, there are genes. That was where we thought we were going to get most of the information. So these are segments that specifically code for something. And oftentimes we'll see, okay, where are the segments that code for this enzyme? Where are the segments that code for this neurotransmitter? We'll talk about the serotonin transporter later. So these are all of the sections that are somewhat related to different conditions. The problem is we thought it was going to be one chromosome, one enzyme for depression or for autism. Yeah, it is not the case. It's a little bit on this one and a little bit over here and something over there. So it was way more complex than what we had originally anticipated. Oops, there we go. So it doesn't mean we can't unlock it. It doesn't mean people aren't still exploring it, but they're exploring it now in a little bit of a different way. So you'll hear these two terms used. They are not interchangeable. They're very different concepts. So pharmacogenetics is only looking at one of those genes. So it's only looking at one small particular point and trying to see if one of those changes makes a difference for patients who may have bipolar disorder, for example. Pharmacogenomics is looking at that whole code, that whole genome, all of the genetic content, because there's little bits on all of those different chromosomes. And if we only are looking for one change, we're likely to miss the picture. We're not likely to be able to pick up some of those bigger signals. So you will see that both of those two terms are used quite a bit, but they mean very different things. How close you're looking versus if you're looking at the whole thing. It's looking specifically at what the genetic code means for how patients are able to tolerate their medications. What would be the most effective for them? What are they going to have side effects with? What might be problematic for them? Ultimately, the goal is to be able to use that information to optimize drug therapy, to have personalized medicine, something that's going to work and that's least likely to cause you adverse effects. We're not quite there yet, but we are on our way. Now, why is that important? Because we can see all of these different changes. For those that are looking specifically at pharmacogenetics, the one gene, they're looking at that whole code, only zooming in on one section, and they're looking for one change. What was the single nucleotide? Was it an A that now is a C? Was it a T that's now a G? They're looking only at that one thing and seeing if that makes a difference. That's going to take a ton of time to work through. And it may not be, that may only make a difference if 2 or 3 other things happen, which is why you'll see a lot of those genome-wide association studies that look at the whole thing to be able to say, okay, well, if there's one change here and there's this change here, now we start to see a difference for our patients. And again, that takes a ton of time to be able to go through. It also requires a lot of information. So we can see some of these differences and things don't happen that we were expecting to happen. So why might that be the case? So if we're expecting something and we don't see it, this was one of the concepts that I think took everybody a little bit off guard. You can have a DNA sequence where it says, okay, well, most people who have this, maybe they have this side effect, maybe they get a rash, but it's not everybody. So it's like, okay, well, we really thought it was due to this genetic sequence, but here's someone who has that code and that's not happening to them. What they found was there is something else going on. They've termed it epigenetics that makes a difference. It essentially can turn some of those genes on or off. Some of it's nature. In some cases, we want our nerve cells up in our brain. We want our muscle cells in our muscles. We want our skin cells in our skin. So all of those cells have all of the genetic information. So in that case, it makes sense that they turned off the liver part for your skin cells that are on the back of your hand. But the environment can also do that. And it can do it in a number of different places. And we weren't quite ready for that because we kind of expected if we see the code, this is what's going to happen. And we've discovered that that's not always the case. There are different lifestyle factors that can play a role. The amount of stress, the amount of sleep, the dietary intake, if people are physically active, things like meditation and prayer, all can have an impact on how much different genes are turned on or turned off. As you would imagine, some of the genes that are involved with things like anxiety, insomnia, those can be turned off with things like meditation, with physical activity, with socialization, with a supportive network. So there's a lot of interest, and you'll see some of the studies. There have been some that have come out over the course of the last about 5 years that are trying to capitalize on that. How can we leverage the environment to also help us with this genetic profile? So that's kind of a wild card that's going to be in the background. We want to focus on how best can we use this profile to care for our patients. So those external factors, yes, we can try to put stuff in place, but the patient ultimately is going to be the one responsible for it. So how can we use the rest of the information? As I mentioned, our ultimate goal is personalizing medicine, saying, okay, for you, this is what would work best. Here's what's not going to give you a bad adverse effect, and it's going to maximize your chances of achieving the goal that you're looking for. Not quite as easy as it looks. So in order to do that, there are a number of different gene labs. You can have a sample of DNA sent. Oftentimes it's a cheek swab. It may be blood work that's sent. Most of your cells have all of that DNA code, so they're able to use any of those cells to be able to get the code. They analyze it through their software, and then most of the reports you're going to see have been color-coded. They often look a little bit like a stoplight. Green tends to be things that are coded as use as directed, not a problem. Then there'll be a yellow column. The yellow column tends to be, there's moderate interactions we may have to use with caution. We need to be careful here. And then there's a red category. So this red category initially was significant concerns. We really need to think about the use of this. They've now renamed it a little bit to significant gene-drug interactions. What is a little confusing is red tends to mean stop. It does not mean that we cannot use the medicines that are in this red category. And I think that's really important to remember. When patients get it and they see the red drugs, they tend to think, okay, no, I'm not supposed to use those. No, we just, again, need to be careful, conscious, and aware of that information so we can use it safely and effectively. This is an example. So GeneSight, I am not a consultant for GeneSight, but they do have an example, a couple example reports that are available on their website. So if you want to look at one in a little bit more detail, this is what it'll look like. So again, you see those red, yellow, and green categories that are listed. And then there'll be a bunch of drugs underneath. If you know that some of your patients or their families have this information, encourage them to share it with the treatment team, because that can open up a discussion. They may be dead set on, you know what, bupropion is in the red category. I'm on it. I don't want to be on it anymore. And that would be a really important discussion to have, because we shouldn't be abruptly stopping many of these medications. Maybe that medicine's working great for you. You don't have side effects from it. Even though it's in the red category, we can continue to use that. But there may be some discomfort, especially when they initially see it here listed in black and white. So recognizing this is how the patients are going to get their information, how that information is going to be shared, let's dig in just very briefly what that information can tell us. So those interactions that they talk about, we're going to focus on the yellow and the red categories. They tend to fall into buckets. So safety, side effect issues, efficacy, how well does a medicine work, and then metabolism. So when you take a medicine, your body processes it to get rid of it. The way it processes it can be impacted by your genetic code. And we'll take a look at one example of that in just a few minutes. Let's start with safety concerns. So the seizure medicines are medicines you sometimes see used for aggression, agitation, bipolar disorder. So carbamazepine or Tegretol, oxcarbazepine, phenytoin, or Dilantin. These medicines have a really bad rash reaction that can happen. They've been able to find that individuals of Asian descent tend to have a genetic profile that increases their risk of developing the really bad reaction. So they can develop something called Stevens-Johnson syndrome. So they can develop a rash that can go head to toe. It can create inflammation and problems with the mucous membranes. It may lead to some of the skin sloughing off, and it can progress to organ damage. It may potentially be lethal. That would be a terrible adverse effect that we're trying to avoid. So for these seizure medicines, it's recommended, it's written right into the prescribing information, that patients have genetic testing done if they are of Asian descent to make sure they don't have this particular genetic profile. If they do, they recommend not using these medicines so we can lower the risk that they'll develop these dermatologic reactions, so the rash reaction and potentially additional damage. So that would be a great thing. This is one of the examples of where it's been able to target therapy. There's no genetic polymorphism. We're good to go. We can use the medicine safely. So it really does take some of the higher risk patients out of that pool. So that's been a benefit. How about looking at the efficacy side of things? How about making the medicine work the way that we want it to? We don't have as much information there. It hasn't panned out quite as closely as we had wanted. There are some examples. I'm going to pick on attention deficit hyperactivity disorder. So there are some genetic profiles where there's changes in some of the dopamine receptors that may impact how well a patient responds to treatment of ADHD. Things like methylphenidate, or the mixed amphetamine salts, so your Ritalin or your Adderall type compounds. It's not consistent, again, because of the epigenetics, but that may guide therapy a little bit to let us know, okay, these medicines may not be incredibly effective. One other example that is being explored really, really heavily is with our antidepressants. So there are genetic profiles that look at the gene that encodes for our serotonin transporter. So we have these little pumps in our brain that grab the neurotransmitters and move it back into the neuron. So when serotonin's out, it can work on other neurons. That transporter pump pulls it back in and says, okay, your actions are done. You're kind of back in timeout. We're putting you back in your room. Well, the gene for that has two different versions, a longer version and a shorter version. They found that people with the shorter version do not make as much of the pump as they're supposed to. Now, that may mean that there's more serotonin available. That may also mean that maybe patients have different sensitivity to that neurotransmitter. It also means that the target for our antidepressants like Prozac, fluoxetine, Zoloft, sertraline, Paxil, or paroxetine, we don't have as many targets for those medicines. So there have been a handful of studies done, very small number of patients that were included, and they've gotten inconsistent results. In some cases, the folks with that short code, they don't seem to respond as well to these antidepressants. But there are other individuals that respond just fine. They're still seeing response to that. So they're looking for, okay, we zoomed in on just this one gene. Is there another signal that can tell us, okay, if a patient has really bad anxiety or really bad depression symptoms, they have this code, we're wasting our time with these medicines. We don't have that yet, but they are very much screening for those type of combinations in patients right now. So it's kind of exciting, not exactly where we want it to be yet, but a work in progress. The last area is probably where we have the most amount of information, and this is on how your body processes medicines. So you take a medicine, it does something to your body. You have a headache, you take some Tylenol or ibuprofen, it helps relieve the pain. That's exactly what you wanted. But maybe it also upsets your belly because of its actions. So the work of the drug on the body is the pharmacodynamic piece. It is not a one-way street because the body also then takes that medicine and is like, we got to break this down and get rid of it. So the way that your body takes in the drug, where it goes in the body and how your body gets rid of it is that second piece. And that's where we see a ton of variability. So we're going to focus just on how the body metabolizes medicine, how it breaks it down and gets rid of it. And your liver is the main workhorse for this. So you may hear people talking about cytochrome P450 or the CYP enzymes. These are essentially the little protein factories that take the drug and they break it down so the body can get rid of it. But genetic differences can vary how effective those enzymes are pretty significantly. One of the main ones is our cytochrome P450 2D6 enzyme. This enzyme is responsible for for breaking down a lot of our medicines that we use for mental health treatment. This particular code is on one of the chromosomes, so chromosome 23, and they've found over 120 different codes. So more than 120 different variables. So not everybody has the exact same breakdown of what that looks like. They've been able to categorize some of the codes. So we have some folks that are poor metabolizers. They have one copy of this enzyme and it doesn't work very well. For those individuals, they can't break down the medicine. These are going to be the medicines that pop up in that red box. It just means we have to use really low doses. There are other folks that may have multiple copies of a really effective enzyme. That means they're going to blow through that medicine. They have lots of enzyme available to chew through this, and we may have to use higher doses for them. Again, they may fall in that red category. Doesn't mean we can't use these medicines. Risperidone is one that's processed by this, but we would just have to be careful and keep that in mind. So we see all of the different profiles. They're trying to identify who fits in each of these, but there is a lot of variability. So this does tend to be one of the areas we focus on most. Does it make a difference? Yes, it actually is resulting in changes in dosing. Some of the examples you can see here, so aripiprazole and brexpiprazole, Abilify and Rexulti, we actually have to cut the dose in half if we know that the patient's a poor metabolizer. Now, if we don't have that information, it just means that patients get side effects on higher doses. We tend to lower the dose anyways. So if we don't have this information, we can still get to the same place, but sometimes it can save us a little bit of time. So that was a lot of information. The slides will be posted for you to be able to go back through, but I really just wanted to do a quick drive-by on this is a field that's changing on a regular basis. The information gives us additional considerations so we can make smarter decisions for the patients, but it is not going to be the solution. It's not going to tell us this is exactly what you have to take and this is exactly what the patient's going to experience, because there really is a lot of internal individual variability. So it's really important to have that dialogue. I made the mistake earlier in my career. Someone who was very excited to get this profile because they're like, this is going to tell me what I'm supposed to take. And I was like, yeah, that's not what that tells you. Please be kinder than what I was in that instance, because they see it as hope. Absolutely, it can give us more information, but we do just want to tender those expectations. Doesn't mean we can't use the red drugs. They may find that the drugs that are in the green category never work for them. And that's okay too. It's just another piece of the puzzle we can use moving forward. So that's what I have for today. Thank you. I will stop my share.